funnel, it's going to drop into one of these kind of like a keyhole or something on the top part of this that acts like a turbine. And as it does that, it causes this top part to move, to gyrate to one direction. And then as this, because it moves, the hydrogen then pushes over a slot, and it's going to fall out this other funnel-like shape over here. But since they're progressively entering this funnel here, they enter and bind. That causes a little bit of torsion, and it falls out here. And this occurs progressively. And that's how this thing rotates like a turbine, and it's just ultimately that, and the energy of that motion that is used to drive this phosphorylation of ADP to ATP. So this diagram gives you a good overview of the process. They say I'm not going to have you go through it. Photosynthesis, we went through a few more of the proteins, but here we're not going to go through the proteins involved here. But you see here that NADH enters in the first protein subunit, whereas FADH2 is only entering here at the second protein subunit. And there are 3 proton pumps associated with complexes 1, 3, and 4. So that's why there are 4 protein complexes and 3 proton pumps. FADH2 is going to come in at protein complex 2, but it's not associated with a proton pump.
Is FADH2 also pumping 2?
It's also pumping 2. FADH2 also carries 2 electrons, and it's going to be pumping, it will cause the motion of 2 hydrogens from inside to outside.
So each flow, the NADH
will bring 6, and it will bring 4. Yeah. So during cellular respiration, most energy flows in the sequence from glucose to NADH, going through the electron transport chain, and this drives proton motive force, those proton pumps, and then ultimately those proton motive forces used to drive the synthesis of ATP. It's not, of course, a completely efficient reaction, as nothing is, but about 34% of the energy in each glucose molecule is going to be ultimately used to synthesize ATP. And as I said, that number is variable, and I'll explain why in a moment. The number is variable from 30 to 34 ATP are ultimately produced. So the reason why the number is variable will be described here. And as I mentioned, this picture I have from the 9th edition, and in this last phase here, they have 26 to 28 ATP being produced per glucose in oxidative phosphorylation, whereas in the previous edition, if you have the previous edition, I think they have 28 to 30 or 30 to 32 here. It's quite a bit higher number. So I'm not going to quiz you on that number per se, but I will talk about what I want you to understand about the production of ATP. Remember, in the first step, in glycolysis and in citric acid cycle, we are producing 2 ATP by a different mechanism, not chemiosmosis. How are we producing those ATP? Substrate level. So the number that we produce in those two phases is known. It's repeatable. And it's always 2, you're getting 2 ATP generated by substrate level phosphorylation. Remember, in glycolysis, you have to input a little bit of energy before you get out 4 ATP, but your net is 2. But it's a constant number that is generated by substrate level phosphorylation. So those ATP will now, those 4 are a set number. But the number of ATP that are generated during oxidative phosphorylation is always given as a variable number and has been difficult to quantify because there's not a direct relationship between how many hydrogen are pumped in and the ultimate number you get through ATP, through chemiosmosis. And there's a couple of reasons for that. The first and most important I'll describe right now. So remember, in glycolysis, we produce 2 molecules of NADH, and NADH is our electron carrier. But NADH cannot, as I mentioned, enter directly into the mitochondria. So in fact, what happens is that NADH comes to the cell membrane of the mitochondria, and he's met by one of two people. You can think of him as meeting one of two butlers, as Kent, one of the other professors says, and I kind of like his use here. So when NADH comes to the cell membrane, he meets either the senior butler, who's NADH, or he meets the junior butler. And when he gets to the door, the senior butler is a little bit more efficient. That's why he's NADH, and the junior butler is FADH2. But either one of them are equally likely to come to the door. So both of these carrier molecules are going to show up with equal frequency. He himself cannot enter, but he can pass on his electrons. So the NADH does not enter, but he can pass on his electrons to either NADH or to FADH2. So he, at this, instead of there being a transport protein of some kind that allows the NADH to come in, what happens is there's an electron shuttle at the cell membrane. So all the NADH is doing is taking that carrier of two electrons, which we've spoken about a few times, as this little courier package. He passes on that courier package with two electrons to either the senior or the junior butler. If he passes it to the senior butler, he's passing it to another NADH, and when it goes to oxidative phosphorylation, it's going to be associated with approximately how many hydrogens being pumped across? Approximately 6. Because it's got 2 electrons that go through 3 proton pumps, and each time they pump 2. So when he comes to the door, you get 6 protons. But when the junior butler comes, you are getting, he is entering, he is passing on his electrons into the second protein subunit, and you're getting, passing through 2 proton pumps, and ultimately getting 4 hydrogens. That's the main reason why you always see this differing, this number differing here by a couple. And it's because it truly does differ. If NADH passes those electrons to either NADH or FADH2 with equal frequency. And when this occurs, you get 2/3 the number of hydrogens. You're getting 2 fewer hydrogens going across. And then the other reason is that it's actually still not known precisely the relationship between how many hydrogens are passed and how many ATPs that you get for that. It's still not completely a known number. And though they try to do it experimentally, I suspect that it has to change based on absolute flow of hydrogens as well. There's many different things, not only this process, which are going to be affecting the hydrogen concentration inside and outside the cell. And it's ultimately that electrochemical gradient which is driving the motion of the ATP synthase. So it must be dependent somewhat on background levels as well. And so probably in recent years they've done more detailed experiments and basically decreased the number from what you'd see in textbooks for decades before that. It now comes out a little bit lower level. But the main point to take home is that one of the reasons, well, there's 2 things. One is that 4 of the ATP are generated by substrate level phosphorylation, and that's a fixed number. Then the second thing to take home is that one of the reasons why there's variation in the number that comes from chemiosmosis is that the 2 NADH that are produced during glycolysis enter the cell and are transferred, the electrons enter and are transferred to either NADH or FADH2. Okay, those are the 2 things I want you to remember about that. Then the conclusion of all of this is that for each glucose molecule, you're getting 30 to 32 ATP. So this diagram here is showing you the amount per glucose. Remember, 2 pyruvate are produced for each glucose. So each of these is going around twice for each glucose. But they're showing you the net numbers here. Okay? Any questions about the number of ATPs produced or how ATP is produced? Okay. So as I mentioned, if you do not, yeah.
Sorry, so for every 1.5 hydrogens?
Yeah, I want to verify the next class because I wrote the number down. I want to verify the next class. So it's about, so here we have, it would be too many? Well, I was thinking for every for every 6 hydrogens, you're getting between 2 and 3 ATP. For every, and that's true. So let's write it that way. I remember that correctly. For every 6, for every 6 H plus, you get 2 to 3 ATP. That makes sense. Yeah, that's correct. Because you have at least 8 NADH here, and sometimes 2 there. Yeah, that's correct. Okay, so for every 6 hydrogens, you're getting 2 to 3 ATP. So as I mentioned, if you do not have oxygen at the end of your at the last step of electron transport chain, this system is going to get bogged down because you're not passing on your electrons. You're not getting, you're filling up your proteins with electrons and they cannot pass them on. So what happens in those circumstances is that, as we mentioned before, the cell does not require oxygen to perform glycolysis. Glycolysis is an anaerobic process. And we spoke about a couple of times now that our very first photosynthetic organisms, like purple sulfur bacteria, they used photosystem 1, and they used the sugar for photosystem 1 to put it into glycolysis, and they got out pyruvate, but they don't use citric acid cycle or oxidophosphorylation. They do not have mitochondria, purple sulfur bacteria. They only have a chloroplast. So these purple sulfur bacteria are generating all of the ATP that they will use by substrate level phosphorylation in glycolysis. Well, it turns out that we can do that too. We don't get a lot of ATP that way, but of course there are times that metazoan organisms are under a state of low oxidation, oxygen depletion. And when they're in those circumstances, they still need to produce a minimum amount of ATP to stay alive. And that ATP is produced by a process known as fermentation. I will go through these. I'll come back to anaerobic fermentation in a moment. So the 2 processes by which fermentation are well known to occur one is called alcohol fermentation, and the other is lactic acid fermentation. In both of these situations, your cells are going through glycolysis. Glycolysis does not change in fermentation. It's the exact same process we saw before. What was that process? We have to use 2 ATP to start the system. We get out 4 ATP by substrate level phosphorylation. So our net ATP is 2. We have 2 ATP as our net. And what's our other product? It is pyruvate, right? So we're getting 2 molecules of pyruvate and 2 ATP for every glucose. All of the metabolic steps are the same as we covered before. But now, what else is produced during glycolysis? Remember, in addition to 2 ATP, we get 2 NADH. Remember, under aerobic respiration, these 2 NADH are the ones that have to go to the cell membrane and meet the senior or junior butler, okay? But in anaerobic conditions, these NADH are not going to the cell membrane, and they are not going to meet any butler. They are building up inside the cell. So if you have a fixed quantity of NAD+, your ATP that is produced is going to go off immediately, be used for cellular work, and it will be regenerated to ADP, no problem. There's all sorts of uses for ATP. But the role of NADH was only to be an electron carrier, right? So if it is not passing on its electrons, NADH is just sitting around now with its basket full and nobody to hand them to them. So the only deal with fermentation is that these are processes by which NADH can be reconverted to NAD so glycolysis continue. In the case of alcohol fermentation, that process generates ethanol alcohol as a byproduct. It's a waste product for these cells. They don't need the alcohol. We like the alcohol. We'll talk about that in a moment. But these yeast don't need that alcohol. It is a waste product for them. And the only reason they put up with it is because they want this guy. They need to recover their electron carrier so they can go through glycolysis again. The same deal is happening with lactic acid fermentation. Glycolysis occurs exactly the same way. You get 2 ATP. And you develop the pyruvate. But now, again, you have your NADH electron carriers filled up after some time of this occurring. All of your NAD plus molecules are going to be occupied. So you have to remove those electrons in order to recover your electron carrier. In the case of lactic acid fermentation, what happens is the pyruvate passes its electrons to lactate and then restores the NAD plus, having it